Thanks for tuning in again for our daily conversation about the latest science involving the COVID-19 coronavirus outbreak. So one of the things that's really been in the news, can't escape it, is the issue of testing. One of the biggest problems that we've had in terms of understanding how many people are affected by this disease is the fact that testing takes some time and we just don't have the capacity to test as many people as we'd like to. But what I wanted to talk about today, and we can kind of get back to how the science might help us solve some of these problems at the end of our conversation today, is how this test works and how that influences the rate at which we can test people and the capacity that we have to run a certain number of tests every day. So the most common test that's being run today is actually trying to look for the presence of the virus itself in a patient. And this is called a molecular test. So we talked on our conversation yesterday about the structure of the virus, about how it's a shell with these spike proteins sticking out of it, and that shell encompasses where the RNA that encodes the viral genome is inside. So that's our squiggly little red line here. Now, the process of doing this kind of molecular diagnostic testing that's being done all around the world is to first extract the RNA, to isolate the RNA from the virus, and then actually convert the RNA, which is a single-stranded molecule, into its matching double-stranded DNA. So now we have the same sequence, but now as double-stranded DNA. Once you have that double-stranded DNA, you can find regions of that DNA that can help us identify two characteristic regions of the protein that we want to identify as a tracer for the virus. So there are two regions in the genome that are usually used. You always want to have two because you want to have one as your primary screen and one as a backup that you want to confirm that both are present to ensure that you're getting the right result. So you have these two regions of the genome that you're trying to amplify. You want to make lots and lots of copies of it because when you're just using, when you're starting with very small samples of the RNA that you might pull out of, a patient's swab that's been swabbed from their nose or their mouth, you're starting with a very small amount of RNA. So to be able to detect it, we have to really make a lot more copies of the DNA before we're able to tell whether that DNA is present or absent. So there's a process called RT-PCR that essentially amplifies each region by duplicating it over time. So every cycle doubles the number of copies of these two regions that you will detect in the sample. So you do that over and over and over again, and as you look at those amplification cycles over time, you see this growth in the number of copies that you have of your target regions. And for a patient who tests positive, you'll see this curve go up over time. If you don't see those, then you can assume that the patient does not have the virus. So this is the basics of the test that's being used right now. But the problem is that even though it's very sensitive, you can start with very little RNA and still get a positive result. The problem is that it requires this sophisticated laboratory equipment to do this amplification process. And so that's one of the bottlenecks in the process, is that there are only a certain limited number of labs around the country that have the equipment to be able to do this. So some people have been talking about a different approach to doing identification of infected patients. And this approach, instead of trying to find the virus itself, involves trying to, let's get that there, trying to look for the antibody response that a patient's body would generate as it's responding to an infection. So it's a little bit more of an indirect diagnosis. And the way that this works here is that, again, let's look at the viral structure again. So we've got our shell, we've got the spike proteins. And in fact, what researchers have found are that the body, when it's infected, can generate antibodies. These are molecules that recognize foreign molecules, that they produce antibodies that are actually recognizing those spike proteins. So that's been documented in patients who have recovered from COVID-19. And so if we want to try and find these antibodies in the body as kind of a marker that the patient has had the infection and then recovered, we can actually use a kind of molecular sandwich of molecules. So what scientists are working on right now is to take the sequences of these spike proteins and turn them into synthetic versions. So these are just sequences that we can produce in the lab, and you can attach them to any kind of a surface like a cell culture dish, something like that. And then run that patient's blood sample. So this time, instead of a throat swab, we're looking at maybe like a finger prick of blood. If you extract the blood sample and you run it against this synthetic spike protein, then if those antibodies are present in the blood sample, they're going to attach to the spike proteins. Now, the second part of the sandwich, the top piece of bread then, is a generic antibody that then recognizes the common section of the human antibody right here. And those generic antibodies are attached to some kind of reporter molecule, something that might be fluorescent or something that gives off a color reaction. And I've indicated those with these little stars there. So essentially what happens in this kind of a test is that the antibodies that we form as an immune response to the virus ends up being this bridge between the synthetic viral proteins and the detection molecules that we use. If you don't have these, then no sandwich forms and there's no color reaction. Now, the problem with this kind of a test is that it is, again, that indirect test. It often takes a couple of days for this response to gear up enough so that we can detect these kinds of antibodies through this kind of test. So it's not as accurate necessarily as the molecular test. So when people these are currently not yet approved in the US for use with patients yet. The FDA has given the green light for the development of these tests. But the idea is that this kind of test might be more useful as a much more widespread test to find out how many people in the population might have had the infection after the fact. So we've talked a lot about people who might have gotten infected but either show mild symptoms or don't show any symptoms. They're not the ones who are going to get the prescription to get the molecular test right now. But this kind of a test, because it's much faster and it's much cheaper, we could potentially administer this test at scale to get a sense of how many people are getting infected after they've already recovered. So the molecular test that we have right now, and then this, what we call a serological test, analyzing what's in the blood, are kind of complementary in terms of what they do, and they have different pros and cons. What's really exciting is that now scientists are really developing new technologies that kind of combine the best of both worlds. So you may have heard in the news about an emerging biotechnology called CRISPR gene editing. And in the case that we're most familiar with, we hear about CRISPR as this essentially molecular cut and paste to edit genes that might be faulty. So maybe curing a genetic disease that has no cure by cutting out the mutated version and replacing it with the correct version. But in fact, CRISPR has actually a much broader application if you think about its key role as being like a search and find function. So what scientists are doing now is applying that search and find function to taking a different approach to developing a molecular test. So in this new approach, which is really just coming out of the lab right now, they're not even into clinical trials yet. This is really just proof of concept right now. Is again, you're starting with the RNA, you're pulling out the RNA just as we did before. And again, you're converting that RNA into that double-stranded DNA again. But this time, instead of having to find these target regions and do all of these rounds of amplification that takes time and equipment, if you use a CRISPR molecule, I'm going to use my little orange block right here to represent our CRISPR molecule. What this block has is a search and find function that can recognize that target region of the DNA that focuses on one of our recognition sites for the viral protein. And what's cool about what's built into this protein complex is that it has a guide sequence that finds the right place on the DNA. And it also has these molecular scissors, as we call the CRISPR Cas12 complex here. And these particular scissors, molecular scissors, are really interesting because they automatically chop single-stranded DNA. So remember, we've got double-stranded DNA here. So the clever thing that scientists have done is to use a single strand of DNA to attach, again, some kind of reporter molecule. Again, could be something that's fluorescent, could be something that creates a color reaction. And so by using this, instead of having to go through this whole amplification, you put this CRISPR complex there, it finds the right part of the DNA, and when it binds, it chops off that reporter molecule. So you're able to get the really fast color reaction that we were talking about as an advantage from the antibody test, but you're getting the accuracy of the molecular recognition of the RNA viral test. So this is, I think, a really exciting new development in how we're taking the best of emerging technologies and applying it to developing faster and better tests for an emerging crisis. So we'll see how this kind of technology progresses. It is not yet approved by the FDA, even with any kind of emergency use protocol. But I think there's a lot of promise here in terms of very rapid development because it uses a lot of off-the-shelf materials. So you don't need specialized labs to do it. It can be cheap. It's fast. The process as it's been right now can take around just 40 minutes. So I think, we'll see more of this kind of technology being used to develop diagnostic tests, not just for COVID-19, but for other diseases as well. So we'll see how this changes the landscape in the months moving forward. Already we're seeing that with the need for more testing, we're broadening the criteria that we have to start testing people. As more and more of any of these tests come online, we'll see much more broader screening. But I think the scientific story here is actually a really exciting new development. So thanks for joining us today. We'll have a lot of fun tomorrow. We've had a lot of questions come in over the course of the week. So we're doing kind of a Q&A with the most frequently asked questions to address some of the ideas that people are seeing in the headlines and maybe some misconceptions as well. So I hope you'll join us again tomorrow. Thanks.